So before I start, I want to thank the organizers to invite me and to present at an in-person conference, which is very exciting. Haven't been doing this in a long time, and it's particularly exciting for me because I'm a very active person. I need to move when I talk, which is difficult in front of a computer in the office. So I will be covering a lot of the stage. For the people online, that's I'm sorry in advance. And I'm particularly excited to be in Nottingham. My kids were even more excited about me being here because they have a very simple connection to Nottingham because of Robin Hood. And the only thing my daughter told me was I should stay away from the sheriff, which I usually try with police. Wherever I go, I try to stay away from them. Okay, but since we are a little bit short on time, I will actually start talking about science. And as you can see on the title, I selected a title that is Cryo-electron tomography workflows to extract quantitative and geometrical information from variable protein assemblies. And yes, that sounds very vague. And the reason for that is because when I had to select the title, I was not really sure yet what I would be talking about. And that is because we work on two main biological topics in the lab. And the one is regulation of the cell motility machinery. So how do cells use their cellular components to move from point A to point B? And the other topic is the conservation and diversity of viral assembly mechanisms with a focus on retroviruses. And what connects these two topics is that when we try to understand the biological mechanisms behind them is that we don't look at proteins in isolation, but we want to look how proteins act together in time and space to fulfill their biological function. And specifically, we're interested in these biological mechanisms where the geometry and the topology of proteins that play a role are contained. This information about geometry and topology is containing relevant information to tell us how the proteins do their function. And this is essentially very similar to what also Julia has just shown you. I mean, it was great that she had given the talk now because I can spare you with all the basics about the method. And extracting this geometrical information and to connect it with functional data is what we need to develop workflows for, because for many things that we want to do, like also Julia already emphasized, there's unfortunately not a one, straight pipeline to do this, but you always have to think for each sample how you go upon it. And since there's not unlimited time, I decided to talk today about our cell biological topic, which is the cellular structure biology of the actin cytoskeleton in cell migration. And I can't spare you biological detail, unfortunately. But I will try to keep it brief and simple. So in general, we want to understand how is the cell motility machinery regulated. So how is a cell able to move in a directional fashion? And the best example to illustrate the question behind this is probably this video that many of you might know, which is from the 1950s or 60s, which shows an immune cell chasing a pathogen. And every time the pathogen is changing its direction, the immune cell, of course, has to do the same. And this is done via polymerization of its so-called actin cytoskeleton to push the leading edge, which is the front of the cell, in the direction that the cell wants to go. And this needs to be, of course, tightly regulated. Otherwise, the cell would be pushing in all different directions, and then it could eventually engulf this poor pathogen that was trying to flee. But resistance in this case was, of course, futile. So yes, I'm a sci-fi fan as well. So what actually is underlying machinery behind this is the actin cytoskeleton, where you have a polymerization of actin monomers into filaments, and these filaments provide the protrusive force to push, but also provide a scaffold for the cell to maintain shape. Now, the regulation of this motility machinery happens on several layers that one has to consider when one wants to understand how it works. One has to look at the morphology and dynamics. One wants to look, of course, how does the cell change its shape, and how do the dynamics of does the dynamic of the proteins change when the cell moves fast, slow, changes direction. One then needs to look at the ultrastructure, where the geometry and the topology of the actin cytoskeleton is always adapting to how the cell moves. You can have bundled filament networks, you can have branched filament networks, and of course, how these networks look like determines what the cell can do. And then you look at the finest level of regulation, the structure and the biochemistry, where essentially actin binding proteins define the identity of a specific actin filament. So, for example, I will be talking about the alpha-3 complex, which you can see here on the right side, that generates branch networks that then eventually leads to cell migration characteristics which are specific. And one could think this is a bottom-up regulation, but it is actually not, because not only the proteins define how the network will look like, but the network itself, the geometry and the producer forces will define the activity of the protein. That means you cannot just look at one thing to understand how it works, but you should integrate information from all of it. So what we do is we use an integrated cellular structural biology approach in the lab, where we use cryo-electron tomography to obtain structures of proteins in cells and also more information about their molecular stoichiometry and the geometry they form. And then we contextualize the structural information and also structural information with cell biology-based approaches to understand the dynamics of proteins, cell migration, and we also generate, for example, knockout cells to understand more what happens when we remove a specific protein. And we do this on many proteins, and I just will talk about one protein where I show you the workflow, and this is the upto3 complex. Long story short, the upto3 complex is extremely important to nucleate new filaments. They don't like to do this by themselves, so it's providing essentially a Kickstarter. It plays a role in many physiological, but also pathological processes, and it generates so-called branched actin networks, where the upto3 complex links an existing mother filament to the growth of a new daughter filament on its site. The upto3 complex is a heptamer complex, 7 subunits, 240 kilodaltons, so it's not massive in size, but it's also not too small. And what happens is that upon activation, the complex needs to change its conformation because it will bind a pre-existing filament to form a so-called branch junction, which nucleates a new filament. And since the fun thing about this complex is it can nucleate its own substrate, it could potentially lead to exponential growth of actin filaments, which is of course not desired by the cell. So it's tightly controlled by many activators, inhibitors, and stabilizers. And understanding how these work, one requires understanding of the structure of the branch junction. So structurally, people have been trying to work on this for many years, and people solved already in the early 2000s the inactive complex via crystallography, which did not tell much about the activation and how it actually nucleates filaments, but it gave very nice insights into the fold of the individual proteins. But what people then tried to do was to reconstitute networks in vitro to use tomography to solve the structure. And this is what people did, for example, in 2008 in the lab of Dori Tanain. In negative stain tomography, they solved a 30 angstrom resolution structure, which gave a rough idea of how it looks like, but that of course was not good enough to really understand more. And people since then have been trying to solve the structure of the branch junction, the activated complex. But in vitro, you can guess from this image, there's a preferred orientation problem when you usually do this because you always look at the complex from one side. People could not push it. So we said in the lab, okay, we are interested in cell migration, we're interested in this complex. Let's try to solve the structure in the cell because we should be able to overcome some of the limitations in vitro. But we're of course expecting some of the limitations you have when working with cells. And the thing is that we have the optimal sample to look at actin networks and the proteins within them, because we look at migrating cells, such as this melanoma cell, which is expressing fluorescent actin. And you can see here that the actin is polymerizing at their very edge. In so-called lamellipodia forming branched actin networks. And these lamellipodia are perfect for electron tomography because they are only 200 nanometer thin, full with actin. So you don't have to do any fib milling, you don't have to do any symovis or anything like that. You just can acquire tomograms. And they're full of branched actin. So perfect. So the first thing we had to do is we had to actually get good sample. And if anyone of you has worked with adherent cells and cell culture, trying to get them onto the grid and do what you want them to do. This is notoriously tedious, especially with these migrating cells. You look at them in the wrong way and they shrink and they don't form any migration protrusions. So 2 people in my lab said, okay, they don't come from the electron microscopy field, but from cell biology. It is like, oh, this is not really what we signed up for. Can we make this a bit easier? So they designed these 3D printed grid holders, which you can print in every, normal 3D printer, to a design that you actually like. You can really easily change this and use them as an adapter to handle grids, to culture cells on them in whatever way you want. And also over a couple of weeks, for example, we do experiments where we culture cells on grids for several weeks and they are fine. And we made these grid holes also compatible with micro-patterning, for example, so that we can actually control where we seed the cells. And that worked actually quite well. So we do all kind of experiments when we culture the cell in discrete holders, and you can see, for example, the micro patterning on the side. So if you're interested, you can download these 3D prints, the STL files, can change them, and you can use them in the lab. So we do essentially all our cell culturing. Now, once we had figured out how to actually get good cells, we chose a cell model. In this case, it's 3T3 fibroblast cells, not important for this year. And we were seeding them on the grid. And what we did now, we do not perfectly work in situ, but we mildly extract and fix them because we thought, okay, we want to start off easy by increase contrast a little bit without altering the network topology and also the structure of the protein, we hope. And we did some controls that I don't have time to show you, like verifying the proteins are still retained. And then we zoomed in on these lamellipodia, which are spreading nicely over holes, as you can see here, where we have still very nice contrast, and then acquired cryo-electron tomography data. And this is what you can see here. This is a standard tomogram we acquire. These black lines, you can see tons of actin filaments. Looks a bit chaotic. It is actually. And if you then know what you look for, you can find these branch junctions quite nicely, actually. So you see here an actin mother filament running from the top to the bottom, and then branching off on the left side, an actin daughter filament. In the middle is the alpha-3 complex, linking together both. So we said, good, we have to find those now. And we can, of course, manually pick. We had 150 tomograms. My postdoc was not excited. So we said, okay, let's try template matching. And that worked reasonably well after cleaning out by classification afterwards. And you can see here some examples of these branch junctions. So we had then our data. We didn't have a workflow, and this is exactly what Julia mentioned before, is you really have to think how you process the data, because you have to use whatever software that makes it work for you. So we started off with CTF-4, Nova CTF. We then used Dynamo for template matching classification. And then we did first alignment. We went to warp and M. We went to Reliant. We went to warp and M again. We went to Reliant. We went to warp and M again. In the end, we got a structure. And of course, this is something that we ended up with after many rounds of testing and really straining our computer cluster to the edge. And we got a 9-angstrom structure of the alpha-3 complex in cells. And that was good, because at 9 angstrom, of course, it's not single particle resolution, but it allows us to fit the crystal structures, the inactive subunits. And that was accurate enough to build now a resolution, a model that is precise up to 9 angstrom resolution of how the complex looks in its active conformation when it's actually nucleating new filaments. And here, this is just a video to show you some of the structural details to convince you that it's actually 9 angstrom. So let me let that run through for a second. So we, of course, learned biological information from that, essentially how the complex changes its conformation from inactive to active. And we could show that there are essentially two subcomplexes rotating against each other. But that story has been published. I won't go into this detail, but we wanted to follow up on this now. I said, okay, we can determine the structure up to 9 angstrom in the cell. Can we learn more about the complex? And the alpha-3 complex is actually not a uniform entity, but it's actually a some of its subunits consist in isoform-specific fashions. So there are in total 8 different variations of how the alpha-3 complex can assemble itself. So we said, okay, how do now these isoforms convey different activity of the complex and also cell migration characteristics? And can we study this from the cellular to the structural level? So to really understand how the change, sorry, I shouldn't move with the microphone away from my face, how the change of protein activity changes topology, changes protein structure and cell migration characteristics. So we generated knockout cell lines where we removed individual isoforms. And you can see here 2 isoform knockouts. I'm not going to bore you with which isoform. And you can see, depending on which isoform is knocked out and which one is now incorporated into the complex, you can see clear changes in the cell migration characteristics and also in the morphology of the cell. While in one the lamellipodium shrinks, in the other one is actually enhanced. So we wanted to understand how is the dynamics changed. So we did all these measurements on polymerization speed, turnover of the network. We also looked at localization of actin binding proteins, but then of course said, okay, now on the ultrastructure level, what's going on? And this is where we face now the problem. How do you go from this nice data to quantitative information that describes it? Because this is just gray value data, but you have to somehow vectorize that information. And you have to then take this vectorized information and compare it across data sets. And we tried around with neural networks to segment this data, which you can see here. Super chaotic, works very well though. But one of the postdocs in the lab was working on another topic. He said, wow, this is too tedious. We don't have the tools. So he teamed up with collaborators, mathematicians, to look into his project, where he works on different actin binding proteins, where we generated knockouts, to understand how the quantitative aspects, the variables that determine cell migration have changed. So they developed the toolbox, which allows you to come in with vectorized information. And here we use tools, for example, that have been developed by the Baumeister lab or by another lab in Vienna who are also mathematicians, to take this vectorized information. So essentially all you get for filaments is XYZ positions, and then display them in whatever way you want, analyze them whatever way you want with predefined parameters that we selected based on what do we want to understand based on the network, and then also classify, for example. And the good thing is you can do this across large data sets and also compare genotypes from different knockout cells. And this is just an example where we compare 2 different bundled filament networks, where we essentially have these parameters, and we can easily spot now what are the differences. We can display them differently, we can analyze them further. And what we can also do is we can actually use information to understand how filaments are oriented with relation to each other, how they are distanced, how they are in angles to each other, to eventually pipe this into processing pipelines later on to use this as a classification tool of geometry, where the geometry, as I mentioned, tells function, which means it must be related to the presence of specific proteins. So this is something that we want to go to. So we did this for these isoforms of the Aptas V complex. We can see clearly that there are changes in the angular distribution patterns. We just display this in different ways now. But we also wanted to check, do the isoforms do change just the topology, also the nucleation activity. So we did template matching in a controlled way to see how many branch junctions do we actually have in there. And we could see that although the network has changed, the branch junction number is actually not. So we want to see what's going on with the branch junctions. So we solved 3 structures for isoform-specific branch junctions again in cells to higher resolution than we did previously. We here acquired around 250 tomograms or so. And we could see that in one of the isoforms, the stability, one of the subunits is really changed, changing the stability of the complex itself, and therefore potentially altering also how the network is stabilized. So you can see this here in this green little rectangle. It's one of the subunits that is different, which is also visible in this difference map that we calculated, where the UpC1 subunit, which is not the one that was actually an isoform knockout, really changes stability depending on what's incorporated into the complex. And this is really nice information for us because it really allows us to integrate now different things. It allows us with our workflow to integrate the cell biological parameters, dynamics, change of polymerization, change of protrusion efficiency, of essentially what defines how a cell moves, with the underlying topological changes, and the structural changes. So here we can link, and this is unpublished, but we're currently writing it up. There's a lot of biology to learn, but I won't go into detail about this, how one isoform really allows cells to migrate. And this is important because the isoforms that we look at actually have been implicated that one is upregulated in cancer, and we have actually now pretty good understanding why the upregulation of one in cancer might lead to enhanced cell migration characteristics, and can link this from the cellular to the structural level. Now, we wanted to see how far we can go in resolution with the branch junctions. We pooled all the data together, knowing that they're not really isoform homogeneous, because we have knockout 1, knockout 2, and 2 wild type, and we got a 6.8 angstrom resolution structure from around 35,000 subvolumes, which means we are still not dataset size limited. We can really go higher in resolution by adding in more data, and that's what we are currently trying to do, because obviously the goal would be to go beyond, let's say, 4.5 angstrom. This is going to be requiring a little bit more of data acquisition, a little bit more of the refinement of our image processing workflows, but I'm quite realistic that we can achieve it. You know, a 6.8 angstrom, that's a nice resolution, but compared to a 7% of an angstrom resolution, you don't learn much, right? Some of the things might be better defined, but beyond, you know, hunting for the number, this didn't show us much. We don't wanted to know how good is our data really, and we tried then to look at actin, because we have a lot of actin filaments in our data. And we used, again, warp and rely, and we got the 3.9 angstrom structure of F-actin within cells, which is now at a resolution that really allows us to see, you know, proper structural detail. We can see the alpha helical pitch, we can see side chain densities, and we can see also, you know, the density for the nucleotide. That is very important in actin filament, you know, elongation and also depolymerization. And that shows us two things. Our data is good enough to go to high resolution if we have sufficient amounts of data, but it also opens up a lot of doors for us now, because with the 3.9 angstrom structure of the F-actin filament in cells, we are anyway focusing on other actin binding proteins. We can now classify on specific actin binding proteins to understand their binding pattern to F-actin in cells. That's essentially all I wanted to talk about. I was speeding up a little bit, you might have noticed. So I want to just quickly summarize again what I showed you. Essentially, I tried to convince you that we have a workflow in-house that really allows us to get decent quality data of our cellular model systems to understand questions on the cell biology side, but also on the structural side, and that we really try to integrate them, because that is telling us most about how the mechanisms work. That we could get a sub-nanometer structure of the activated up-to-3 complex in the branch junction, and in the meantime, I've refined it a little bit. We are currently trying to push it further, but that is probably a story in 2 years or so. That we are trying to do this integrated cell and structure biology on isoform-specific knockouts, where, you know, the advantage of cryo-electron tomography is really that you can now use genetically altered cells to understand within a cellular context, not only structure, but also all the things that are related to the change of, you know, the genotype. And that we, you know, doing further developments, I did not have time. I actually have a bioinformatician in the lab working on really implementing new processing workflows for F-actin to get better structures of F-actin from lamellipodial actin networks for, you know, more complicated biological questions in that context. Now at the end, I want to you know, acknowledge everybody who has been working on this. So this is the group at ISD Austria, and I mentioned we work on structural virology as well. Unfortunately, I couldn't talk about this, and you have to believe me that I find this equally interesting. And the work that I've shown you here today was mostly done by Florian Schur, a postdoc in the lab, Bettina Zenz, a PhD student, and Georgi Dimchev, who was a postdoc but has in the meantime moved on. And I also want to thank collaborations we worked with, specifically Clemens Rottner and Martin Falke. And you for your attention. I'm happy to take any questions.